What's up, YouTube? Sorry, I always wanted to say that. Okay, so in this chapter 14, we're going to be going over the nervous system. So a couple of things I want to point out before we actually dive into the nervous system itself is that this is one of my favorite, actually my favorite organ system. I actually did my graduate work on the nervous system, particularly the nervous system of Drosophila melanogaster, or fruit fly. So I get really excited about this. B, this is a very, very complex organ system, and it has a lot of complex physiology or function that's happening with it. So there are going to be lots of things that are going to seem like you're not getting the full story and the full pictures, because you're not. We're getting like the tip of the iceberg, if you will. This is very much an overview and an introduction to this very complex organ system. As I said before, you can actually take entire year-long classes on just the brain itself, just one organ of this organ system. So just to give you a picture of idea of the complexity. So there will be some things that we don't dive into a whole lot of depth on, because you just don't have the background knowledge for everything that's going to be covered. C, there will be some parts that we cover in this lecture that you might not necessarily see in your book, or there might be parts in your book that are expanded upon more than we have in this lecture. And the reason being is that I like to keep these lectures within 45 minutes, 30 to 45 minutes, and not too much longer than that. So I won't be going over every single detail in this chapter. All right, so without further ado, let's go ahead and get started. So an overview of the nervous system is that its basic job is to receive and process sensory information from both the outside world and the inside world. So we can take that receiving and processing information and then coordinating the output, and we can separate it into two different entities. So the two major divisions of the nervous system are those that consist of your brain and your spinal cord, the central nervous system, and then you have the nerves, which are consist of the peripheral nervous system. So the nervous system has three basic major functions. A, it's sensory. That means that it needs to pick up the sensation of stimuli either outside of the body, i.e. the stove is hot, the wall is gray, or sensation on the inside of the body. Smooth muscle contractions of the uterus signaling that a baby needs to come out, or smooth muscle contractions of the uterus signaling that endometrial wall needs to come out during your menstrual cycle. So it needs to receive the sensory information, and then it's going to send that information via the peripheral nervous system, the stove is hot, through the nerves to the central nervous system, the brain, and coordinate a response. Integration is where the central nervous system, brain and spinal cord, basically sum up what the appropriate response is to whatever this internal or external stimuli is. So whether it's moving your hand, or looking more at the wall because you like that it's gray, or whatever it may be. Now, the motor portion of this, so sensory is the sensory information or the signal coming into the central nervous system. Motor integration is saying, what should we do with this information? And then the motor response is going to be how your muscles respond. So going back to the stove is hot, you sense the stove is hot, peripheral nerves tell you to your brain and your spinal cord. Your brain and your spinal cord says, well, move your hand. And then the brain and the spinal cord say, move your hand. It's going to send that signal out to your muscles that say muscles contract to move your hand away from that stove. So that is the motor portion of it, or the motor output, which travels from the central nervous system to the peripheral nervous system, either the muscles to act a certain way, or the glands, or even other organs to coordinate their activities. So when we look at nervous tissue, all of the nervous tissue that we're talking about, peripheral nervous system, all those nerves, brain and spinal cord, they're really only made of two different types of cells. You have neurons, which their whole job is to send and receive signals, and then you have neuroglia. The neuroglia, or the neural glia, their whole job is to be like the supporting cast members of the nervous system. Now, think about it this way. So let's use Kim Kardashian. So Kim Kardashian is like the star, okay, of her own little reality world. So Kim Kardashian is the star. But you realize that she doesn't wake up looking like that. Don't get me wrong, she is an absolutely beautiful woman. But She doesn't wake up with full face of makeup and full hair done and fully dressed. She wakes up just like the rest of us do. She has really bad breath, I'm sure, in the morning, because everyone has really bad breath in the morning. Her hair is probably disheveled. She doesn't have any makeup on. So she has a team of people to give us the brand Kim Kardashian. So she has a stylist. She probably has multiple stylists. She has makeup artists. She has hair artists. She has a whole team of people that support her brand. So in this case, the neurons would be Kim Kardashian, and the neuroglial cells, the ones that support, would be all of her supporting cast members, the people that actually help her do her thing. So in just the same way that the cast members or the people that actually help make the Kim K brand, there are more of them than there are Kim Kardashian. The same thing with the neuroglial cells. They're going to greatly outnumber the number of neurons that you have because they have very specific jobs that they do. So some basic neuroglial cells that we're going to talk about in the central nervous system are microglial cells, which have an immune response, so they fight bacteria, astrocytes, which help to maintain the blood-brain barrier, and oligodendrocytes that help to make the myelin sheath that goes around the nerves. Now, the peripheral nerves, the peripheral system has its own class of neuroglial cells, and those would be just Schwann cells, and those Schwann cells help to make the myelin sheath, or the part around the nerves in the peripheral nervous system. So microglial cells, as I said before, they have an immune response. Astrocytes, they help to maintain that blood-brain barrier, so they do so by providing the metabolic and structural support to the neurons. Oligodendrocytes form the myelin sheath in the central nervous system, and they're called Schwann cells that form the myelin sheath in the peripheral nervous system. So now that we've talked about the basic types of neurons, now we're going to talk a little bit about, well, when we say basic types of neurons, we're talking about between the nerve cells that we find in the nervous system, between neurons and the neuroglia. Let's talk about some very basic types of neurons based on their function. Neurons can be sensory neurons, as we saw, for the sensory information needs to come into the nervous system. So remember we talked about how the nervous system has three functions: sensory, integration, and motor. There are specialized neurons for sensory information, carry signals from the nerve or the sensory receptor to the central nervous system. So let's take your eyes, for example. Your eyes don't actually see squat. I'm just going to throw that out there. Your eyes are sensory organs, which means that their whole job is to perceive or to actually give you this image or whatever it is in front of you, but it doesn't actually see anything. You can't take your eyes and separate them from your brain and then just think they're going to walk around and start seeing stuff. They see because they are connected to your brain. It is your brain, your occipital lobe, that actually translate those images into some sort of picture that makes sense for you, that makes that picture clear. Now, don't get me wrong, there are conditions like cataracts, there can be scarring or abrasions of the cornea, that very first part of your eye, that can occlude your vision, that make it very difficult for you to see what it is that you should be perceiving, but it is your brain that actually perceives that. So the sensory neurons, your eyes in the retina, they have a bunch of these bipolar sensory neurons, and they're called those cones and rods. Those cones and rods are what perceive color, what perceive shape, what perceive anything that you're looking at in your external world. But there is a sensory nerve, so those sensory neurons are then connected to that optic nerve that goes straight to your brain. So those sensory receptors, bipolar photoreceptors in your eye, that detect changes in light and changes in the environment. So then we have interneurons. Interneurons are only found in your brain and your spinal cord. So whenever you see C and S, I want you to think brain and spinal cord. So they take the input of that sensory information. So since we're using the eyes, let's keep with that. They take that input information from the eyes and other things that the other different neurons are giving us, and then it sums up the signals that are received to communicate with the motor neurons to say, hey, this is how I need you to respond to this particular sensory information. And so then the motor neurons, the last type, their job is to carry the nerve impulse, or whatever the command was from the brain and the spinal cord, to the effector. So that could be a muscle fiber, contraction of the muscle, it can be an organ, the eyeballs, or it can be even a gland, the pancreas gland, the pineal gland, any of those. And that's going to carry out the response to whatever change that happens there. So notice how these three types of neurons directly correspond to the three functions of the nervous system. So for any of those three neurons, they all have three distinct parts to them. Now, there are going to be lots of other parts that your textbook talks about that are found in neurons, but they're not always found in all neurons all the time. These three parts are found in all neurons all the time. So they all have a cell body, which contains the nucleus and all the other organelles that we've talked about. Dendrites are the short extensions of the cell body, and their whole job is to receive sensory information or to receive signals from other neurons. So the dendrites are the receiving end of the neuron. The axon, on the other hand, is the sending portion of the neuron. So that's going to be the portion that conducts the nerve impulse or that sends that message of what should be happening to the effectors or the glands or the muscles or the even other neurons. So dendrites receive the message, axons are going to send it. Individual neurons are termed nerve fibers, and collectively all of those nerve fibers form what we call a nerve. So when we send that signal off, there are a couple of ways that we can do it. We can just send it directly off that axon, and it's just what we call continuous propagation, and that we have an action potential, which we'll talk about how an action potential is generated in just a few slides here. We can have that action potential happen all along the length of that neuron until it gets to the next cell that it needs to send that message to. Or we can speed that process up. So if we have continuous propagation, which means that an action potential happens on every single portion of that axon, that happens a little bit slower than if we can get the action potential to jump from one part of the axon to the next part of the axon to the third part. So that instead of having to have 15 action potentials along the length of the axon, you only have to have, let's say 4, because it jumps from one node to the next. So you may be asking yourself, well, Kel, how does that jumping take place? Why didn't it take place on the continuous axon where it didn't have, where was it going? What was the difference there? The difference is a myelin sheath. So remember, in the central nervous system, the myelin sheath is composed of the oligodendrocytes, and in the peripheral nervous system, it's made out of these Schwann cells, and those are both the supporting cast members of the neurons. So the myelin sheath covers portions of that axon, that part that's supposed to send the message. And the parts that aren't covered, the action potential can jump from one of those parts not covered to the next part that's not covered. And those are called the nodes of Ravnier. So those are just the spaces between the myelin sheaths. So because it can jump from one space to the next, it can happen much faster than if you have to have an action potential on every single part of the neuron. So case in point, the Hulk. He can basically jump from one place to the next. If the Hulk is jumping from this area to the next area and to the third area, he's going to get to that area a whole lot faster than somebody that has to run or fly that entire distance, because he can just jump over those portions that they have to fly through. So nodes around near are those spaces between the myelin sheath where the action potential is generated. So long axons usually don't have a myelin sheath. The long axons usually have a myelin sheath where short axons don't. And that's just because there's more distance to be covered. There's gray matter of the central nervous system, and the reason it's gray is because there aren't any myelinated axons. And then there's white matter, and that's usually found on the outside. So gray matter is usually on the inside of the central nervous system, so the inside of the brain or the inside of the spinal cord. And then white matter is white because the Schwann cells or the oligodendrocytes actually give that myelination a white sheen to it. For people that have multiple sclerosis, that is where the myelin breaks down. So the neurons aren't able to transmit information either at all or not as quickly as they did before. So this myelination is important, as you can see, because we talked about multiple sclerosis being a condition where there's not enough myelination of those nerves. It's important also to help regenerate within the peripheral nervous system. So when you have nerve damage to any of those nerves, the myelin sheath, those Schwann cells, can actually help to regenerate the nerve itself. If the axon is severed, the myelin sheath remains and acts as a passageway for the new fiber to grow. So that's how that process takes place. So now for the next couple of slides here, we're going to talk about that whole action potential thing that needs to go down. So there are a couple of slides that seem like they're going to be a little fuzzy, and I apologize for that. So for this slide, and there are quite a few more, this next section we're going to talk about is how does a nerve cell do its job? What is its physiology, or how does it function? So a little bit of vocabulary here. Nerve signals are really electrochemical changes that send information within the nervous system. Remember, nerves don't tweet, they don't text, they don't Facebook, they don't Snapchat. So in order for them to send information, it has to happen on a chemical level. It happens specifically on an electrical chemical level. So that means there's going to be voltage change, and that voltage change is going to be brought about as a result of negative ions, negative chemicals, negatively charged chemicals, and positively charged chemicals. Resting potential is the voltage or the electrochemical number when a nerve isn't doing anything, or actually not just a nerve, but when any cell isn't doing anything. So when a muscle is at rest, it has a certain voltage to it. When a nerve cell is at rest, it has a certain voltage to it. Anything that has to do some sort of job has a resting potential. And that resting potential, because it is a voltage, it's measured in millivolts. Resting potential of every cell is about negative 70 millivolts. So I want you to remember that number, negative 70 millivolts, because we're going to see it quite a bit. And the reason that we have this resting potential is because usually there is a more positive charge outside of the cell than there is on the inside of the cell. There are more negatively charged entities, more negatively charged chemicals and ions on the inside of the cell. So when a cell is not doing anything, it's just negative. So negative 70 millivolts is the resting potential of pretty much every cell, specifically your nerve cell and your muscle cell. So if we were to take a voltage meter and measure the potential of this cell that's at rest, we would see that the inside of that cell is at negative 70 millivolts, and then the outside of that cell is more positively charged. So inside, if we were to look and see what makes this negative 70 and what makes this outside more positive, we would see inside we have lots of these little chemicals, and we have some of them are positively charged, the potassium and the sodium, and then we'll have others that we're not actually looking at right now that are more negatively charged. So then we have these channels that these sodium and these potassium ions can go through. So more resting potentials. Neurons have to maintain this resting potential in order to send nerve signals. So you have to be down at negative 70 millivolts in order to generate an action potential. Action potentials happen at positive 30 millivolts. So in order to get to positive 30, you need to be negative first, right? Or you have to be less than at least positive 30. So that's why we need this negative resting potential. In order to go from a negative number to a positive number, are protein carriers that do this. And we have something called the sodium-potassium pump that will pump sodium and potassium out and into the neuron in order to change that resting potential. So what I want you to start seeing here is that the resting potential was caused by the number of positive and negative entities both inside and outside of the cell. Inside the cell, there are more negative things than there are positive things. There are more positive things on the outside of the cell. There are some negative things on the inside of the cell, and the ones that we're mostly concerned with are sodium and potassium. If we move the amount of sodium and potassium that are inside the cell, we can go from a negative number to a positive number because both sodium and potassium carry a positive charge. So if we were to open up those gates and bring more positively charged sodium ions into that cell, it now goes from negative 70 to like negative 60, negative 50, negative 10, positive 10, positive 20, positive 30, or an action potential. The more positive things that you bring into the cell, the more likely we are to have an action potential. And that is the neuron during work, action potential. So nerve signals are also called action potentials, what we've really been talking about here. Resting potential is what again? Negative 70 millivolts. Action potentials, we want that to happen at about plus 30. So you haven't seen that number yet, but it's coming. So a stimulus is some sort of change that activates that neuron. It could be a prick if it's a pain neuron that senses it. It could be lights if it's photoreceptors that sense it. It could be all sorts of things. It could be a chemical, acetylcholine, that causes the muscle cells to now start to generate an action potential. Now, the stimulus, like I said, is anything that changes that neuron. Threshold is the minimum amount of voltage change that has to happen in order for an action potential to take place. Now, once we reach threshold, that means that boom, we're going to have an action potential no matter what happens. It's an all or nothing sort of response. So it's not like you get to negative 50 or negative 45 and you're just like, I'm just going to have like a little bit of an action potential. No, once you reach threshold at negative 55 millivolts or about negative 55 millivolts, that signal is going to be sent. You can think of it this way. If you put your finger on the trigger of a gun, after you put so much pressure on it that you've reached threshold, and that you put enough pressure on it, that's going to trigger the hammer to be released. It's going to hit that bullet out of the chamber, and that bullet's going to go out at the same velocity as if you didn't put as much pressure on it. So you can't just say, oh, I just squeezed the gun a little bit, the trigger a little bit, officer. The bullet should have just come out really slowly. No, once you reach threshold and put enough pressure on there, that bullet is going to come racing out of the chamber just as quickly as if you would have just, pulled it very quickly. So that's the same thing that happens with action potentials. Once we reach threshold or this minimal voltage change, we then will get an action potential to take place. So here's just a really cool picture showing that we get up to about negative 55. We're getting more of these positive entities. Notice that we have these channels opening up. So we're getting more sodium in the cell. So we're going to get more of these positively charged entities into this negative area that took us from negative 70 millivolts to about negative 55. So now that we've reached threshold, we are ready to have this all or nothing action potential. So once it's reached, the action potential happens. Doesn't vary in size, like that velocity of that bullet. It didn't slow down because you just barely squeezed the trigger. It didn't speed up because you squeezed the trigger very aggressively. So they happen. They're what we consider all or nothing. So instead, to convey a stronger signal, it causes more action potentials to happen. So you get more action potentials to go on. So that's what that process consists of. So now that we have this action potential, we can call it an action potential, or what we can call it is depolarization. So when the action potential begins, protein channels open up and allow sodium, or those positively charged entities, into the cell. Adding these positively charged entities inside the cell causes the action potential to take place. So there are more positive things coming in. The axon changes from a negative to a more positive. And now we have depolarization. When you see depolarization, I want you to think action potential. When you think action potential, I want you to think positive 35 or plus 30 millivolts. Now, what goes up must come down, right? So after we've had an action potential generated, we need to bring it back down to resting potential so we can have another action potential generate. So immediately after depolarization, aka action potential, aka positive 30 millivolts, the sodium channels close and then the potassium channels open. So when we have those potassium channels open now, so now the sodium channels are closed, no more sodium is getting in, but we have those potassium channels that are opening up, and we're kicking all of these positively charged potassium ions out. So we keep kicking these positively charged potassium ions out until the inside of the cell becomes negative again. Finally, at the very end of this, we have a special pump called the sodium potassium pump that completes the action potential. So the sodium are returned to the outside of the cell, and sodium or potassium is returned to the inside of the cell and sodium to the outside, and we get that resting potential restored. So just what I want you to remember about this is that the sodium potassium pump helps to restore that resting potential. And here is repolarization. So we've been looking at this resting potential, negative 70 millivolts, depolarization, positive 35 millivolts, and then going back down to resting potential, just in what's happening along the length of the axon. We can actually chart that on a graph. And when we chart that on a graph, it looks something like this. Down here at resting potential, reached up the threshold, we're going to have the action potential, depolarization happens, sodium moves inside the axon, repolarization happens once we get it to about plus 35. We want those sodium ion channels to close. Potassium ion channels will open up. We kick potassium out. We keep kicking potassium out, keep kicking it out until we get down here to resting potential. And then we have that sodium potassium pump that helps to maintain us at negative 70 millivolts. So when we move that action potential, remember that there were two ways that we said we can do it. On continuously along the axon, which means that that whole process has to happen on every single part of the axon, or we can do it by jumping from one node to the next, and that was with that myelination. So we can have continuous propagation or movement of that axon. If there's no myelin sheet, it takes a little bit longer, or we can have the myelin sheet where the action potential, that whole song and dance that we talked about with sodium coming in, hitting up positive 30, and then we get to positive 35, and we want to kick potassium out, and then we want that sodium potassium pump to reestablish that resting membrane potential. That whole process can happen either along the entire length of the axon or just on specific points of it. So there are just specific points that it can happen. If it just happens on specific points because it jumps from one node to the next, those nodes of Ravnier, then that process will go faster. And that's basically just what this slide says because you can't really read any of the words. So what we call this jumping from one node to the next is we call it saltatory conduction or saltatory propagation. So we have action potential happening here, we have it happening here, we have it happening here, but we don't have it on these parts where the myelination is taking place. So it's literally jumping from one node to the next. So when an action potential goes, we already talked about it goes, it's off to the races. It either happens or it doesn't happen. So we have these periods or these time frames around the action potential. Refractory period is that period of time immediately after an action potential during which the axon is unable to conduct another action potential. So you can think about it this way. We're already at positive 30 or positive 20. There's no really place for it to go because we already have the maximum amount of sodium ions to elicit that electrical chemical change that we want. We already have a bunch of those positive things in there. So you're not going to get another action potential on top of another action potential. And we call that a refractory period. So we have to wait until that action potential is finished till we get down to at least like negative 60, negative 65, and then we get another action potential. So because of that, it makes sure that the signal is always going to go in one direction along the length of the axon, and that we don't have signals getting stuck on the axon, if you will, or going back and forth between the cell body and between the axon, and they're just continuously sending that message in one direction. So a couple of parts of the axon itself. So we talked about the length of the axon is where that action potential takes place, either at nodes of Ravnier if it's myelinated, or along the entire length of the axon if it's unmyelinated. If it's continuous along the length of the axon, if it's saltatory, that means it jumps from one node to the next. At the very end of that axon, you have these little fine extensions that we call axon terminals. So each one of these little fine extension is either very close to either the dendrite or the receiving end of another nerve cell, or it's very close to the cell body of another nerve cell, or these axon terminals can be very close to another type of cell, like a muscle cell. That region, they don't quite touch each other, they just have a little space. That space is what we call the synapse and the synaptic cleft. So the synaptic cleft is just a small gap between the sending neuron and the receiving neuron. And it's a little bit of a misnomer for us to just say receiving neuron because can be any type of receiving cell. Neurotransmitters are specific types of chemicals that can be released from the sending cell that will trigger the receiving cell to also generate action potentials. So remember, cells don't Facebook, Snapchat, tweet, or do any other of those forms of communication. They communicate via chemicals. So a neurotransmitter, a chemical that can go across that synaptic cleft, bind to the receiving end of that cell, and open up those sodium-gated channels so that those positively charged sodium ions can come into that resting receiving cell in order for it to receive the message. So events or things that happen at a synapse, the nerve signal travels along the axon, having those action potentials that take place until it reaches the axon terminal. Once it gets to the axon terminal, calcium channels are going to open up. When those calcium channels open up, calcium is going to go into the terminal and it's going to stimulate those vesicles that contain that neurotransmitter to be released. When those vesicles are released, those neurotransmitters are going to go across the synaptic cleft and bind to the membrane of the receiving cell, and they bind to special protein receptors. When they bind to those receiving cell receptors, then it's going to allow for those positively charged sodium ions to come into this resting receiving cell in order for it to also generate an action potential. And then here is just a super duper cool picture of that exact thing happening. So let's blow up a little bit here so we can see things a little bit nicer. So we have this action potential that has come down. Now that we have reached the end, the action potential signals for these calcium channels to open up. Once calcium comes in, it triggers the release of the neurotransmitters that are found in these vesicles. So they are going to diffuse across this space between the receiving end or the shipping end and the receiving end. So this guy's sending the signal, he's receiving it. And then when they bind, let's go a little bit down further, those little blue jobbies are the neurotransmitters, and they're going to bind to the receptors on the receiving neuron. When they do so, they're going to allow for, you guessed it, sodium ions to now come in. When those sodium ions come in, then it's going to now change this resting cell to a action potential, and so that it's going to have another action potential begin over there. So because of these different types of neurons, it can either, we've just seen in the example, open the sodium channels to excite that receiving cell so that it has another action potential, but sometimes the neurotransmitter can be inhibitory, which means that it causes potassium ions to leave that receiving cell so that it really stays negative. So if it's exciting, that means that sodium ions are coming in. So excitation is where the neurotransmitter causes sodium gates to open, or inhibition is just where the neurotransmitter, what's released, causes potassium ions to leave out of the cell so that we don't generate an action potential. In fact, we get further away in inhibition from receiving an action potential. So whether it's excitation or inhibition that takes place, that's completely determined on the type of neurotransmitter. The response of the receiving neuron can be toward excitation or toward inhibition, depending on what signal they received. So once that neurotransmitter, whether it's an inhibitor or an exciter, once it's been received, that message has been received, we need to get rid of that neurotransmitter. So after it's done its whole job, there are enzymes that will actually break down that neurotransmitter, and one of those such enzymes is acetylcholine esterase. Now, one of these terms you're very familiar with, acetylcholine, because we talked about that in the muscle notes, where we said acetylcholine will bind to the receptors on the muscle cell, allows for sodium ions to come in, and that's that action potential that takes place. What acetylcholine esterase is, and just a little FYI, for the most part, in biology, anything that ends in ASE is usually an enzyme. So acetylcholine esterase is the enzyme that will break down that acetylcholine that's left over in the synaptic cleft. Because remember, we can't have another action potential happening while there's still an action potential doing its thing. That we had that refractory period. So acetylcholine esterase breaks down that acetylcholine. And other types of synapses, the sending membrane can actually absorb some of the neurotransmitters. So there are a couple of different ways that we can remove this neurotransmitter from the synaptic cleft. So acetylcholine is just one neurotransmitter. There are more than 100 neurotransmitters, and we don't know. And when I say we, I'm talking about the scientific medical research community. We still don't know exactly 100% what every last one of those neurotransmitters does. I'm just going to be honest. The ones that we are very intimately familiar with are acetylcholine, norepinephrine, dopamine, serotonin, glutamate, and GABA. So those are the ones that we are very, very familiar with. The rest of them, we don't really know exactly what they do, but we do know when we've done research that if we remove these different neurotransmitters, the nervous system and other parts of the body don't really function like they should. So we do know they're important. We just don't know how they're important. So nerve muscle, nerve organ, nerve gland synapses also use these same neurotransmitters. So even though we're just talking the nervous system, as we've seen acetylcholine in the muscular system, these neurotransmitters can be sent and receive messages all throughout the body. Both acetylcholine and norepinephrine are found in both the central nervous system and the peripheral nervous system. Acetylcholine in the peripheral nervous system acts as neuromuscular junctions. Norepinephrine also has a very similar role. So in the peripheral nervous system, acetylcholine excites skeletal muscle. However, point I want to make is that the neurotransmitter is the neurotransmitter. It just sends the message. How that receiving cell responds, that's a different story. So in the peripheral nervous system, acetylcholine excites the skeletal muscle, but it inhibits the cardiac muscle. So when there's acetylcholine that's released at cardiac muscle, those cells have a different response to that message. Norepinephrine excites smooth muscle. In the central nervous system, norepinephrine is important for dreaming, waking, and mood sensations. Serotonin is involved in your thermal regulation, sleeping emotions, and perception. And then there are many drugs that act in the synapses, like illicit drugs or even pharmaceutical drugs, that can interfere with how those neurotransmitters are doing their job. So integration is really just the summing up of all of these excitatory and inhibitory signals that are coming into this nerve. So if a nerve cell was to receive more excitatory signals than inhibitory ones, then an action potential will be generated. On the other hand, if there are more inhibitory signals sent than there are excitatory signals, then it's going to have more of an inhibitory response. And what I mean by that is that when we look at a receiving cell, it's not just like one, it's communicating with just one cell at a time, although we've been kind of talking about it that way. There are multiple different nerves and axons that are connected to these cells. So there are a whole lot of signals that are being received. So how this receiving cell receives them and what it decides to do, that just depends on what is the sum total of excitatory and inhibitory signals that are received. So if we have here this integration that's going to be determined, like I said, if we have more of these, or if it's inhibitory, then we have these excitatory signals, then we can get an action potential, or we could just not get one. So it's an integration of both what's coming into that receiving cell. So now we're going to talk about, now that we talked about the physiology of the nervous system and how it does its thing, the last little bit here that we're going to go over, this next section, is going to be going over the parts of the brain and the really parts of the brain and spinal cord. Central nervous system, brain and spinal cord, receives sensory information, initiates the motor response. Both the brain and the spinal cord are going to be protected by your vertebrae and your skull. The brain and the spinal cord have special coverings that wrap around them to help protect them. Those are called minnings. And if those minnings ever become infected, which it can happen, but you do have the astrocytes and you do have that blood-brain barrier that's maintained to keep pathogens from getting into that area, and microglial cells that are supposed to help to kill off pathogens. Whenever we say pathogens, remember we're talking about bacteria or anything that causes an infection. If that can cross the blood-brain barrier, it can cause meningitis, which can be caused by bacteria or viruses, and it can be fatal. So I'm pretty sure you guys have heard of different meningitis that have been fatal. So there are two types of nervous tissue in the central nervous system, the gray matter and the white matter. Whether it's gray or white depends on whether or not it's myelinated. Gray matter contains cell bodies and really short non-myelinated axons. White matter has myelinated axons that move in tracks that we call, or bundles that we call tracks. So white matter found on the outside of the spinal cord, gray matter kind of in the inside butterfly portion of it. Brain is made up of these big portions called the cerebrum, the diencephalon, the cerebellum, and the brainstem. First, let's hit the cerebrum. The cerebrum also has another name called the telencephalon. It's the largest part of the brain. This is a part that is the last center to receive any of that sensory information and to carry out that integration before voluntary motor responses. So this is a part of your brain that says, oh, I received the signal that this stove is hot. I need to move my hand from this hot stove. All right, I'm going to take the signal out to the nerves that go to the muscles of my hand that will remove my hand away, or the muscles of my biceps really, to move my hand away. The cerebrum also coordinates and communicates with the activities of the diencephalon, the cerebellum, and the brainstem, so these other portions of it. So this is the most evolved part of your brain. So their thinking happens here, memories are stored here, understanding complex things, problem solving, this is all found in different parts of your cerebrum. So here we have that cerebrum. Let's get a little bit closer so we can see some of this. We have that cerebrum, we have these meninges that are on the outside. On the inside, we have this tract called the corpus callosum that connects your left hemisphere with your right hemisphere. In any of these ventricles, the lateral ventricle, the third ventricle, the fourth ventricle, their whole job is to make sure that the brain is bathed in cerebral spinal fluid that helps to carry nutrients and whatnot. The diencephalon is going to be what surrounds the thalamus. And at the base of the thalamus, you have the hypothalamus, which connects to this hypothalamus here, which there are different hormonal responses, which we'll talk about in chapter 16 in a little bit. You have your pituitary gland that's also here in your brain. And then we have our brain stem at the base, our medulla oblongata, so this is the pons, this is the medulla oblongata, and then we have the cerebellum, and then more ventricles, and we have a here that connects from the brain, specifically from the least evolved portion of your brain, which is your medulla oblongata, down to your spinal cord. And this is just a really cool picture of what an actual human brain looks like. And although all of this is called cerebrum, there are different portions of your cerebrum that have different jobs. This long line in the middle is what we call a longitudinal fissure. So that divides your left and your right cerebral hemispheres. The way that it communicates with one another is through the corpus callosum. And then you have these folds, these giant folds on top, those are called the gyra, and then the sulci are just the little invaginations or the little indentions or valleys. So those cerebral hemispheres are divided into distinct lobes. So remember, it's all called the cerebellum, but we can divide it into different parts. Frontal lobe is right there in the front. The parietal lobes are right there on the side. The occipital lobe is the one in the back, and the temporal lobes are right there above your ears. So we have frontal, parietal, or parietal here, occipital, and temporal. Now, depending on what portion of the brain we're at, they all have different jobs. So the frontal lobe is where you have your primary motor, you have your premotor area, you have your speech area in your frontal lobe, and you have what's called your prefrontal cortex. This is actually the last part of your brain to develop, and actually in humans, prefrontal cortex does not really completely finish developing until you're about 25 years old. So you're still having brain developing happening, which explains why young people do such idiotic things. Their brain is literally not developed. So that happens in your prefrontal cortex. In your parietal lobe, you have what's called primary somatosensory information. So the coordination or processing of information that comes from your the external stimuli and the sensations that come in there. It's kind of the coordination with that. Your taste area is in that area in your parietal lobe as well. General interpretive area, Wernicke's area, where you are interpreting speech or understanding what this speech means, or what, like what is the context of this. So that's a general interpretation area. To smell, that's right, the bottom of the frontal lobe. Temporal lobe, auditory association, which makes sense because your ears are right there. Sensory area, Wernicke's area, that's also very near the general interpretive area. And your occipital lobe, primary vision, and the visual association area. So the optic nerve would go directly from your eyeballs, which is about right here, to the back of your brain. And that's where you actually see. That is where that vision is perceived of what it is that you're looking at. So if there is pressure on that optic nerve, you're not able to actually see, because there is physiological damage that's happening. It's not just with a lot of blindness, it's not always just the function of the eyeballs not working. It could be that the nerve itself is damaged, so it's not actually able to generate an action potential in order to translate those images into clear pictures. So primary motor area in the frontal lobe, also voluntary signals for skeletal muscles begin here, muscles that move your face begin here. If you don't have enough oxygen during birth, that could damage this motor area of your primary motor area, and that could result in cerebral palsy. So when we look at this primary motor area in both the red here and in the orange, let's get a little bit closer. Depending on what part of that primary motor area are we what's going to be affected there. So if we're kind of in this back portion of it, the toes and the feet, the legs, thigh, pelvis, just different portions here. As we get to closer to the front of it, facial expressions, that speech area is here, swallowing, and so forth. Now if we go just to the primary somatosensory area, these are places like for your tongue and your pharynx movement, the teeth and gum sensation there, lips and face, trunk, pelvis, thigh. They kind of go back to they kind of mimic the same thing that's on the primary motor area. So right behind that central sulcus of the parietal lobe, you have the primary somatosensory. Sensory information from the skin and skeletal muscles come here. There are large areas that are dedicated to this acute sensation. So the most of what's kind of be the signals that are received are the face and the hands occupy the most area or highest portion on the primary somatosensory area. So you have receptor areas for taste, vision, hearing from any of those sense organs. So primary taste area is in your parietal lobe. Primary visual cortex is in the back of your brain, the back of your head, your occipital lobe. Primary auditory area is in your temporal lobe. Remember, that's the part of your brain right above your ears. And the primary smelling area, or the olfactory area, is in your frontal lobe, because your nose, makes sense, is right under the front part of your brain. So now on to some of these association areas. So in these association areas, this is going to take information that you have processed in, and it's kind of like the filing center of how these pieces all fit together, right? So you see your mother's face, and you have learned from years of knowing that's your mother's face. That information, you see her face, comes into your occipital lobe, and then in these association areas, you associate this face with this term, mother. So it's where that kind of coordination begins. So of course, it makes sense that memories are going to be stored here. And your premotor area, that's the anterior to the primary motor area, that's kind of organize those motor functions. And then somatosensory associated areas are going to be places that process sensory information from your skin and from your muscles. So visual association in the occipital lobe associates new visual information with stored visual memories or recognition, that face recognition. Auditory association, it happens in your temporal lobe, that associates new sounds with other auditory memories and how they relate to one another. So what I want you to really understand from all of this stuff that we're talking about is that you have different parts of your brains that have different jobs and they perceive different information. But in each one of those parts, you have an association area that takes the new information that comes in and it coordinates it with the other information that you already had in a little file cabinet in there, and it helps you to make that association. So it helps your brain to make connections or make new connections with old information that you have. And that is, my friends, what we call learning. You take new information, mix it with the old information, and you're able to formulate what you expect to happen or what should be the case when you merge those two things together. So processing centers, prefrontal areas in the front of the brain, it processes reasoning, planning, action, and critical thinking. Remember I said your prefrontal cortex is not really developed until you're about 25, which is one of the reasons why young people do things without actually planning very well, and they don't really have the same reasoning of full-fledged adults, if you will. The Wernicke's area is in the back part of your temporal lobe that helps for understanding of both written and spoken words. For people that have like dyslexia or any of those dyslexia type learning um, conditions, they sometimes have a difficult time with writing and seeing letters the same way. Their brains just operate a little bit differently. The Wernicke's area is just different. Broca's area directs the primary motor area to stimulate your muscles for actually speaking and for writing. So white matter is myelinated, descending tracks from the primary motor area of the brain to the lower brain centers and sensory information up to the primary somatosensory area. So there's that myelination that takes place. These tracks also take information to and from different sensory and motor association areas. And then that big chunk of white matter that's happening in there, that's at corpus callosum, and that's going to join both the left and the right hemisphere together. Basal nuclei are just masses of gray matter that are within the white matter. They help to integrate motor commands to ensure that the right muscle groups are stimulated or inhibited. Parkinson's disease is caused by a degradation of the neurons that are in that basal nuclei. So because your basal nuclei ensure that movements are coordinated and smooth, if you have Parkinson's disease, you're breaking down those neurons in the basal nuclei. So they have this kind of very shaky movements because they don't have, or these parts that should making sure movements are smooth have been degraded. So let's move on to the next portion of the brain. We have the diencephalon that includes the hypothalamus, thalamus, and the pineal gland. It's going to be surrounded by the third ventricle. Hypothalamus is an integrating signal. Hypothalamus is important for the regulation of hunger, sleep, thirst, body temperature, and water balance. It also controls pituitary gland, which is what we call your master endocrine gland, and it is the primary link between the nervous system and the endocrine system. Thalamus are just two masses of gray matter, receive sensory information except for smell that goes somewhere else, and then it sends it to its appropriate part in the cerebrum. So it's kind of like Grand Central Station, if you will, in that it needs to send things to different parts. And it gets all of this sensory information in. It's like, okay, you need to go here, occipital, you need to go here, hands, you need to go here, whatever it is. Your pineal gland secretes that hormone melatonin, which helps to regulate your daily rhythms, or what we call your circadian rhythms. Cerebellum is under the occipital lobe, separates the brainstem from by the fourth ventricle. It has this white tree-like matter that we call the arbor vitae, and the job of your cerebellum is to maintain posture and balance. When you become drunk, intoxicated, it is your cerebellum that is mostly affected. So people have had heavy amounts of alcohol. That's why they cannot stand upright. It's why they have a poor time maintaining their balance, and that's called ataxia. Going down to the brain stem, the lesser evolved portions of the brain consist of the brainstem, the midbrain, pons, medulla oblongata, and those three. Midbrain is a relay station, the cerebrum and the cerebellum. So there are reflex centers here for visual, auditory, and touch or tactile. Pons communicates between the cerebellum and the rest of the central nervous system. The medulla oblongata regulates heart rate and it regulates breathing. And there are reflex centers that coordinate head movements in response to visual and auditory stimuli. And you can do these things without actually thinking about these are reflexes you don't have to think about. So medulla oblongata, least evolved, and unlike Colonel Sanders, the teacher in Waterboy, if you have not seen that movie, please, by all means, Google Colonel Sanders, Waterboy, or just rent the movie. It's a classic. Medulla oblongata is not why you get so angry, and alligators aren't angry because they got all them teeth. But the medulla oblongata has a very unevolved portion of your brain, and it has reflex centers for regulating your heartbeat, maintaining your blood pressure, and breathing. There are also reflex centers for vomiting, coughing, hiccuping, and swallowing, right above the spinal cord, and it contains tracts that ascend or descend between the spinal cord and the higher brain. So it's like another relay station, but just not as an evolved of a relay station. So medulla oblongata is responsible for lots of really important things that keep you alive. So stuff that you don't have to think about that keeps you alive. In fact, your entire brain stem does these really important things that keep you alive that you don't have to think about, that's involuntarily controlled. That is the reason why if you have a severe injury to your brain stem, those injuries are often instantaneously fatal because there are so many important things that happen at the base of the brain, that brainstem, that, like I said, are responsible for keeping you alive. So the reticular formation is a network of nuclei or masses of gray matter and fibers that extend through the brain system. So we talked about how the different parts of the brains have different jobs, they store different different information, and they integrate that different information, and you have learning that happens. But we want to pull all of this stuff together, and that's the job of the reticular formation. A major component of the reticular activating system, or RAS, arouses the cerebrum and causes a person to be alert. So when you talk about that RAS system, and you may have heard about this before, that helps to make sure that every part of your brain is alert and it is doing the things that it needs to do in order to get the job done. So what your RAS can do is it can filter out unnecessary sensory information. So that is, you can study with the TV on. You can study with music on. You could be deep in thought about a problem and someone could be talking to you. So it makes sure that your alertness and your focus is put in a certain area. We can have an injury, severe injury can cause a coma to your reticular formation. General anesthetics can actually suppress it. So it can actually suppress it from what it needs to do. And although this RAS and the limbic system, they aren't actual direct portions of your brain that we can point out, they are equally as important as any of those areas that we've discussed. So your limbic system integrates emotions with higher mental functions, so reasoning and memory. This includes your amygdala and the hippocampus. The amygdala is the portion of your brain that creates the sensation of fear. So what happens here in this limbic system is that something has happened. It was not a favorable thing that happened. You remember that thing that happened. Um, whether it was, you remember really weird things about it, like the smell of it, the taste, if that was involved, what it felt like. So anything that comes close to feeling like those things or smelling like those things will trigger your amygdala. So that amygdala says, hey, spidey senses are going off in here. We need to get out of this situation because remember when such and such and such, and even if your brain can't recall the exact thing that happened, it knows that something that was similar to this same situation, the same stimuli that are coming in, something similar to that may be happening now. So that's what that sensation of fear is. The hippocampus plays a critical role in learning and memory. So it makes sense that the hippocampus and the amygdala would be a portion of this limbic system because you remember and you've learned, I need not go down that street because this happened to me. I remember what happened the last time, or the last time I smelled this type of smell, there was XYZ that happen. And they can also be good things that happen there, that your hippocampus, and you can actually retrain your brain to associate, to not have that fear trigger so pronounced for that amygdala. So we have these regions of the brain that we talked about. There is your amygdala, and then there is your hippocampus, and they're all very close to this master endocrine organ, the pituitary gland, where there are lots of hormones that are released from here. So it makes sense that, and when we talk about the endocrine system and the hormones that are released there, that these portions of the brain that are responsible for this learning and this limbic system are very close to this hormonal gland. So a few definitions here: memory, ability to recall information from the past, learning to retain and use past memories to make different decisions or make the same decision depending on what it is. Short-term memory stored in the prefrontal area. Long-term memory is memorization of facts. Somatic memory is memory of numbers and words. Episodic memory, memorize persons and events, and skill memory are performed motor activities. So whether you're a wrestler and there's certain moves, or you practice jujitsu and there's certain moves that your body just from doing that skill over and over again, or riding a bike or throwing a ball, any of those things, those are a different type of memory. So there are lots more than one type of memory, and one type of learning there. So long-term memory storage and retrieval. Visual memories are stored in the visual association areas. Sounds are stored in the auditory associated areas. The hippocampus, which remember we talked about that in the limbic system, so this is a bridge between where memories are stored and where memories are used. That amygdala is responsible, as we said before, for fear and conditioning and associating danger with those memories. And it's really just a way of trying to keep you safe, if you will. Let's say, evolutionary speaking, you went down that way, you almost got attacked by a bear. So going down that way again, you don't want to get attacked by a bear, so you have that memory that says, I'm not going down that way anymore because I almost got attacked by a bear. But at the same time, right, your book only talks about the fear aspect of the amygdala, but your amygdala and your limbic system, you can do that to work for you in different ways. That with meditation and with more positive thinking, and there are different types of personal development classes that have these different mantras that you would say to yourself over and over again, that a lot of times in our modern society, we don't really worry about bears attacking us, right? We worry about not having enough money, not getting the job that we want. We have more of these, less there's fear for my life, more of fear for my quality of life, right? So we can do different things to not trigger our amygdala to be so jumpy, and actually have certain situations trigger more positive um, associations with a particular event. So left hemisphere and right hemisphere are two different parts of your cerebrum, and they have quite a little bit different job. Your left hemisphere's basic job is for verbal, logical, and analytical. Your right hemisphere is more nonverbal. It's your artsy-fartsy side. So it's intuitive, it's more creative. On there's a really good book that I'm reading called My Stroke of Insight by a neurologist who actually had a stroke, and she had her stroke in her left hemisphere. So her verbal, logical, analytical portion of her brain had some damage that was done to it. The right side of her brain, which was more creative and intuitive, was perfectly fine. And she talked about how she was able to really see the connections between humans and in life more than she saw it before, when her right side, or right hemisphere, was the more dominant one. All right, so that is the end of the nervous system. When we come back here again, we will talk about the endocrine system and see how the nervous system and the endocrine system do their thing together. Have a happy morning, noon, or night.